Hello, everyone, and welcome to March's Webinar Wednesday. I'm here with our Chief Scientific Officer, David Barron, and our guest presenter, Benjamin Freedman, and we are going to be talking about new gene editing technology, what it can mean for the future of PKD. I want to just remind you, as always, that this webinar is not intended as a substitute for a visit with your physician or healthcare professional. If you have questions, you can, I'm trying to advance, there we go. If you have questions during the talk, you can submit them via the question or chat box on the webinar screen. You can also submit them via Twitter to @PKD Foundation using the hashtag Webinar Wednesday. And please follow along to the Twitter feed. We'll be tweeting throughout the event. And with that, I'm going to go ahead and pass it over to David.
Well, it's my pleasure to introduce Dr. Benjamin Freedman. Dr. Freedman is an assistant professor of medicine at the University of Washington in Seattle. He has traded coasts during his career. He first became acquainted with stem cells and a fascination with them at the University of Pennsylvania as an undergraduate and at UC Berkeley as a Ph.D. student. He was a postdoctoral fellow at Harvard, where he performed pioneering studies using pluripotent stem cells to recreate features of human polycystic kidney disease. The Freedman Laboratory is currently applying the kidney in a dish technology that he's developed to study the mechanisms of PKD, discover new drugs and therapies for PKD, and is innovating new strategies for kidney regeneration. The PKD Foundation has recently funded a project of his modeling human PKD cystogenesis with pluripotent stem cells. And I'm sure Dr. Freedman will define some of those big words. Dr. Freedman, it's all yours.
All right, thank you very much, Dr. Barron, for that great introduction, and Alexis. And it's really my pleasure to talk to you today about new gene editing technology and what it could mean for the future of polycystic kidney disease. So the other day I was passing through the airport in Seattle and I snapped this picture of an airport departures monitor, and beneath it you can see there's an advertisement there for gene editing. So I thought that was fascinating that in such a public place, we're seeing advertising for this scientific technology. And it says gene editing to end cancer. So I figured it would be an interesting topic to discuss and get an idea of what the possibilities are for gene editing for PKD. So today's webinar, we will start with an introduction to genetic disease and gene editing. I will then proceed to give an example of how in our own lab we've used gene editing to recreate PKD in a dish. Then we'll proceed to talk about the future and kind of gaze into the crystal ball of how gene editing might affect therapies and therapy development. And then we can conclude with some Q&A. So let's start with a little intro to genetic disease and gene editing. So genes are actually encoded in our DNA. So what is DNA? DNA is a biomaterial, and it contains 4 possible letters, which we refer to in shorthand as A, T, C, or G. So if you look on the right here, you can see the double helical ladder structure of DNA. And each rung in this ladder is actually composed of 2 of these little letters, and they make special pairs with each other. So the letters here are referring to specific chemicals. C is cytosine, pairs with guanine, which is G, and A pairs with T. And it's really the sequence of these letters in DNA that is important. So a gene is actually a paragraph, so to speak, of these letters, of these 4 letters, in a specific sequence. And what does this paragraph do? Well, each gene encodes a recipe for a specific protein. And proteins are the physical tools that our cells use to do their little jobs. Okay, so that's the idea behind DNA and why it's so important. And in each of our cells, we have in our DNA the information for every gene and protein in our body. So why do we care? Well, many human diseases are actually caused by mutations in our DNA. We know of hundreds, maybe thousands of disorders that can be caused by mutations in single genes. And these are kind of like typos in our DNA. And there's a list here on the right where you can see many of these types of diseases. And obviously one of the most prominent and common is actually polycystic kidney disease. Although on an individual basis, each of these diseases is relatively rare, in aggregate, they affect a lot of people. And in fact, about 1 out of every 20 individuals is estimated to be born with some type of genetic disorder. In addition to these single gene diseases, which are very specific, mutations also contribute to the diseases that affect large portions of our population, such as cancer and heart disease. So really, genetics, we all know, is very important to human disease. And just to focus in a little bit about what we know so far about polycystic kidney disease, we know the genes that cause this disease. So autosomal dominant PKD, which is the most common form, is caused by mutations in either of 2 genes. And these are known as PKD1, and the second gene is PKD2. The more rare autosomal recessive form of PKD, or ARPKD, is caused by mutations in a gene called PKHD1. And these 3 genes encode 3 proteins. And these are depicted here in this review paper. So you can see the structure of these proteins, polycystin-1 or PC1, PC2, and fibrocystin. And together, these 3 proteins make up a little police force of our cells that guards our cells and guards our kidneys against cyst formation. So normally, we know a few things about how this happens. We know that the proteins normally do their job in a specialized antenna of the cell, and this is depicted here in this image, where you can see a cross-section through a kidney tubule. And there's this little antenna that sticks out of each cell. So the cells here, there's several cells that are making up the circle of the tubule. And each one is projecting this little antenna into the inside. And we know that these PKD proteins actually function together. So this green, red, and blue dot are our 3 members of the police force here. And they function together at this antenna-like structure. When you have mutations in these genes, so mutations that cause polycystic kidney disease, result in a broken member of the group. Okay, so one of these proteins is broken. It can't do its normal job, which is to prevent cyst formation. And a couple of years ago, we also showed that if one of the proteins, for example, can't make it into the antenna, the other ones also can have trouble making it into the antenna where they have to do their job. And as a result of this dysfunction of the group, the cells have problems. So they undergo cell division in abnormal ways. They move around abnormally in the tubule, and they may cause imbalances in the accumulation of fluid and its processing through the kidney tubule. And as a result, in specific little spots along the tubule, these cyst structures arise, which are sort of balloon-like. They bud off of the tubule, and there's millions of these little tubules in the kidney, and as a result, you get thousands of these little cysts that over time expand and disrupt the kidney's function. Now, the details of this are not that well understood, and we still need to do a lot of research to understand how these proteins work and why the cysts form when one of the proteins is broken. Now, the ability to edit human DNA would make it possible to better understand and treat disease in general, and PKD potentially in particular. So for research, what we can do is test the effect of genetic mutations if we can edit the DNA. And potentially for therapy, we could actually correct those genetic mutations, knowing what they do in human patients. And depicted here are a couple of cavemen trying to make fire, and that's because we're still very much at the early days of this type of technology development. Editing DNA in human cells has traditionally been very inefficient, and nonspecific. So the reasons for this, if you look at the amount of DNA in a cell, and here what you're looking at on the right is a book where they've printed out the human genome in 5-point font, and you can see these little, hopefully you can see the little letters, the A, C, T, Gs, okay? And this is just one book. The entire genome takes about 40 volumes, or even more than that. So there's really just a tremendous amount of information in our genome of these, and there's only 4 letters. So you can imagine that finding a specific sequence of DNA in all of this is kind of like looking for a needle in a haystack. And this has made it difficult to specifically edit DNA. We can't even find what we're looking for. Furthermore, our bodies have evolved elaborate security systems that protect our DNA from random changes. So DNA is constantly being damaged. It's estimated that about 10,000 damaging events occurs in every cell of our bodies every day. So biology has evolved specific ways to protect ourselves from these mutations. But these are also security systems that we will need to overcome if we are going to edit DNA and correct the few typos that have snuck through. So to address this, scientists are developing new tools that let us edit DNA more specifically and efficiently than ever before. And although we're still, I think, a little bit in the Stone Age, we have seen a gradual progression over the last 30 or 40 years with a series of technologies such as viruses, homologous recombination, which won the Nobel Prize, all the way now to the common era where we're creating these targeted technologies such as TALENs and the newest kid on the block, which is CRISPR. And these are getting progressively more efficient and specific over time. And the point of the webinar today is really to better understand this technology and to think about how it affects human health, and in particular, how it could affect polycystic kidney disease. So just to focus on this CRISPR technology because it's gotten into the news lately, the way CRISPR works is it uses a protein that can search DNA for short words and therefore find a specific sequence. So if you look here at this word processing picture, you can go to the search function, and by typing in letters, we can recognize progressively specific sequences in this DNA sequence, which is actually from the PKD2 gene. And that's basically how CRISPR works. And it lets us home in, just like a search engine would in a word processor, on a specific spot in the human genome. And this greatly increases the specificity and the efficiency of our gene editing. Although I hasten to say that it is still nowhere near 100% efficient. So CRISPR, there are other technologies that can also do similar things as CRISPR, but CRISPR is cheaper and it's very easy and relatively quick to adopt compared to these other methods, so scientists have embraced it. So that's our intro, and now let's take a real-world example from my lab of how we're actually using genome editing to recreate PKD in human mini-kidney organoids. So the premise and focus of our lab is to use human stem cells and organoids to understand and treat disease. And the idea here is a general one, which is that we can now derive stem cells from a patient's own body. And using science, we can take those stem cells and derive from them these little pieces of tissue called organoids. And each of these tiny little organoids has some similarity to an organ in the body. And these are now derived, so we have new tissues that we could not normally make. We need to use science to make these tissues, but they are derived from our own bodies. And we can now use these little organoids or tissues in the lab to study human disease and as surrogates for the patient to perform drug testing. In the long term, we may be able to push this technology forward and actually create not just little organoids, but actually whole organs, which could potentially be transplanted back into the patients, where they would be 100% immunocompatible. So this is obviously a powerful technology, and we're interested in our lab in applying it to the kidney. So for many years, people could make tissues from hearts and liver and other types of organs in the body, but kidney was difficult. But we've had breakthroughs in our lab and a couple other labs around the world over the last several years. And now scientists can actually grow what we call mini kidney organoids from stem cells from people. So here depicted, I want to just show you what an organ looks like here on the left, and one of these little mini kidney organoids on the right. So on the left you have a newborn mouse kidney. This is a scale bar, so it shows that this little white bar is about 1/10 of a millimeter. The whole structure here that you're seeing is about half a kidney. It's about 1 millimeter in diameter, maybe 1.5 millimeters. And hopefully you can see the very beautiful and intricate architecture of the kidney. There's different cell types here shown in different colors. There's these yellow cells and red cells that are forming sort of circles, circular aggregates on the edges of the structure. And these progress into these long tubules into which the urine is actually filtered and collected. So it's really a beautiful structure that we're born with. On the right here is what we've been able to produce from human stem cells. And the stem cells in this case were actually originally derived from a skin cell from a patient. We transformed those into very early embryonic cells and pushed them to try to develop into a little mini kidney. And when we do this and we treat them with the right factors, we can create structures such as this one, again about 1 millimeter in diameter here, and you can hopefully see that there's a certain organization. You've got these clusters here in red on the edges of the structure, which are the filtering cells, and then more internally there is a nest of tubular structures shown in green, and these are actually proximal tubules. So the structure here has some functionality. It has certain features that make us convinced that it is actually a kidney tissue. And hopefully you can see it's somewhat similar to the structure on the left, although there's still a long way to go between a mini kidney organoid and an actual kidney organ. And we do not yet know whether, for example, these can function at all if we were to put them into human patients, and most likely they would not. We still have a long way to go from organoid to organ, but we've come a long way over the last 5 years, and hopefully more progress will be made. So in the meantime, what we can do with these little organoids is we can use them to study the human kidney and human kidney disease. And we started off with a simple question, which is, can we, now that we've made these little organoids, could we potentially try to recreate polycystic kidney disease in a mini kidney organoid? So here you've got your control organoid. We say, what if we introduce mutations that cause polycystic kidney disease? Might we see formation of, for example, a cyst-like structure? And this would give us a miniaturized polycystic kidney in a dish that we could then use to study the disease and try to test out drugs. We wanted to start with a very clean slate genetically to make a very clean comparison. So we turned to gene editing CRISPR technology, and we designed CRISPRs that targeted a specific place in the PKD2 and a gene, and that's shown here as an example. We also did this with PKD1. But let's just take PKD2 as an example here. So this is actually the genetic sequence, this series of little colored peaks. And you can see here in our control, it's red, green, green, blue, blue, blue. These are different letters. And then there's another blue and black, black. And this gray shading here is the place where we targeted the CRISPR. So we were trying, in this case, to disrupt the normal sequence of the gene. And here in the lower segment, you can see the sequence in our PKD2 disrupted mutant line. And it starts off sort of similar to the control. It's red, green, green, blue, blue, blue. But here at this point, the blue and the black overlap. And from that point forward, the peaks get very messy, indicating that there's a mutation here. And at the very end of it, you can see that instead of getting black, green, black, black, green, we're getting green, green, black, green, green, black. So something really has gone wrong at the DNA level. And when we actually look at the protein that is encoded by this DNA, shown here, and we look at this polycystin 2 police force sort of gene, and here this dark band in the wild type or the control shows that the protein is being expressed normally and the cells have it there. In our knockout line, our mutant, there's absolutely no protein being produced. So this member of the police force is essentially disappeared. So we've used this technology for both PKD1 and PKD2 to create gene-edited lines, stem cell lines, with PKD mutations. And now the question is, what would happen if we turned these into organoids? So at first, when we did this, we did this side by side with the wild type controls that don't have the mutations and with the PKD cells that had the mutations, and we turned them into organoids. And we originally observed that the number of organoids per well was very similar between the different lines, indicating there was nothing fundamentally wrong with the lines. These stem cells could produce kidney organoids. And initially, we didn't see much of a difference. So we did what anyone else would do when we don't see a difference. We just sort of waited and let these organoids sit in the dish for about a month. And when we did that, something remarkable happened. We began to see the emergence of these translucent cyst-like structures, specifically in the cultures that had the PKD-mutant organoids. And here's a picture here on the right where you see two different structures. One is the control, where you just see tubules, and one is the PKD structure, where you see tubules, but also the formation of a round structure, which is actually a cyst. And when we quantified this, we could see that there were many more cysts in the PKD cultures compared to the cultures that did not have the PKD mutations. Now, the genetic background for these cultures is the same. The only difference is that we've introduced a specific targeted mutation in the PKD lines. either in PKD1 or in PKD2. So we are convinced that what we've actually done here is recreate cyst formation from PKD tubules. We can now look at these cysts more carefully using microscopy, and when we do this, we can kind of cut a cross-section into them, sort of like you would if you were cutting a racquetball in half here. And you can see that here this large cyst is actually lined with cells on the edges, but is hollow inside. And it's connected here at the bottom to a tubular structure. So we believe it's actually emerging from the tubules. And this gives us a way now to look at PKD with new eyes. So normally cysts actually form from kidney tubules in living animals, maybe in patients. It's very difficult to observe this process. So what we've shown is that using microscopy, we can actually follow individual cysts as they appear. For example, in this movie here at the bottom and in this time-lapse photography at the top, we can see the emergence over a series of several days, a tubular structure which then expands and begins to form a cyst, and then you have a full-blown cyst by day 16. And we're hopeful that this will provide us with insights into how PKD cysts form in people. So with that, I think it's time now to gaze a little bit into the future and use our imaginations. How could gene editing affect the future of PKD, both in research as well as potentially in therapy? So one thing we could do now, potentially using these CRISPR organoids that I just told you about, is to perform clinical trials in a dish for polycystic kidney disease. So what I've shown you here is a little movie of a plate. Each little rectangle here on the plate, each little square is a well that contains cultures of cells, and we can imagine printing, using this kind of robotics, printing these little organoids in each well of this dish. And there's 384 little squares, or 384 wells in each of these dishes. And that means that if we printed organoids in each of them, we could test a different drug in each well and see if it could, for example, rescue cyst formation. So this gives us the ability to perform experiments with huge chemical libraries on a scale that we could not do in patients. And we could test many different drugs. So these types of robots are available to us now, and we can start looking for therapies first in the organoids and then later, hopefully, in patients. Furthermore, I've already introduced the concept of producing organoids and implanting them back into people, but what gene editing lets us do is to perform a genetic correction step at the very early stages. So we can not just introduce mutations, but we could actually correct genetic mutations from the stem cells that we derive from the patients. And then if we were to produce tissues for those patients, the tissues would be gene corrected and they would end up being disease-free, so that the tissues would not, for example, a kidney that we put back into a patient would not then become polycystic. From a research perspective, CRISPR allows us to turn genes on and off. And this is a very powerful tool to let us understand how genes contribute to cyst formation in polycystic kidney disease. As I mentioned before, there's still a lot of fundamental questions. We don't understand how PKD proteins really police cyst formation. There's an entire genome out there of other proteins that we don't understand what role they play in this process. We know that some of them are important for the function of this cellular antenna, but we don't understand how that works either. So what gene editing allows us to do is to create new models of how these genes are operating, so we can get the bigger picture of what's happening in our cells and know how to treat that. And this isn't just limited to human beings or to cells. CRISPR also enables us to create new types of designer animals, and we can now use these to study PKD. So the mouse has really been the sort of workhorse for PKD research for many years, and we've made a lot of great discoveries based on mouse research. And CRISPR has been very successfully applied to mice. And this is going to allow us to create mice with many more mutations than before, with mutations that are specifically found in human patients, so we can test the effect of specific mutations. We can even envision replacing the mouse PKD genes with human versions, human copies of those genes. So we can study human PKD gene function in a small animal like a mouse. It's not just mice. We can also use CRISPR successfully in large animal models that have a veterinary need. Many animals also get PKD. And these animals also more closely resemble human beings. So by developing veterinary therapies in larger animals, genetic models using CRISPR, we can improve our understanding of how to treat this disease in human beings, which are more similar. And here on the right is a pig, and CRISPR has already been used to change the pig genome in many places simultaneously. And the idea here, and this is work done by George Church's lab at Harvard and a Cambridge company devoted to this topic, is to create pigs that are more immunocompatible with human beings by modifying their DNA. And the long-term vision here is that we may actually be able to create human-animal chimeras, which would be animals with people parts or with people DNA in them, that we can use to study DNA and potentially as a source of tissue. Although all of those ideas are still untested and very far away. It should be pointed out that humans or any species will reject the cells of another species very vigorously. So let's also think about how we can use this directly in humans as gene therapy for PKD. Can we gene edit our own cells in living patients? And I think that PKD might be a good candidate for gene therapy, and I'm going to list the reasons here. First of all, PKD is caused by single gene mutations. We know the genes. And this means that we know, in theory, which parts of our DNA need to be corrected. And we now have the technology to correct those parts of the DNA using these gene editing tools. So in theory, it could be a very clean approach. Furthermore, drugs have not yet been proven effective against polycystic kidney disease. There's obviously exciting candidate drugs in the market or going to market, but they are not there yet and they are not fully effective. So perhaps it's time to consider genes as an alternative approach, gene therapy. Furthermore, PKD cyst formation, we know, is a slow and relatively rare event. So we don't need to 100% cure the disease. We just need to control it and control its effects. So here on the right, you've got what I would say is a mature polycystic kidney. Imagine if we could apply gene editing, even if it's not perfectly efficient, if we could get it to slow down the disease in enough cells in the kidney, we might be able to then get a kidney at the same age, but with fewer and less large cysts. And this could provide potentially enough time for the organ to avoid needing renal replacement therapy. And finally, we know that kidney cells actually like to absorb nucleic acids, absorb DNA, so we may be able to use DNA to target this specific organ of the body. All that being said, there are also reasons to curb our enthusiasm here, and I've listed these. Safety, efficacy, and cost remain major challenges to this idea of using gene therapy for polycystic kidney disease. From the efficacy standpoint, gene editing tools are still very far from 100% efficient. So we need to deliver the therapy in an adult patient potentially to billions of cells, and that's akin to replacing every brick in a house, or maybe every brick in the Sears Tower. So, that's a lot of work, and it's going to take a lot of methods development. From a cost perspective, we would need to tailor the therapy to each individual's specific genetic mutation. So everybody with PKD, each family has a specific mutation, but they're not all the same. And we don't know these a priori, so we have to figure out what they are. And sometimes, even if we try to look at the DNA of the patient, we still can't tell whether the mutation is actually causing the disease. And this goes back to the point of that we need better understanding of how genes are actually causing PKD. So the cost here is a significant barrier. And furthermore, from a safety perspective, CRISPR and all these other gene editing tools remain new technologies. They're not 100% specific. So sometimes you're trying to change the PKD gene, you might end up changing a different gene. And if you're doing that with billions of cells, you're running a risk of, for example, changing a gene that could affect, it could make a cell more tumorigenic. So the side effects here could potentially do more harm than good in the patients, and the first rule of any physician is to do no harm. So these are all serious considerations which will need to be worked out first in preclinical models, in organoids, in fish, in mice, and much later be applied, I think, to patients with PKD. At the same time, I think that it's definitely worth thinking about it, because at the root of all of PKD is a genetic problem, and we might be able to use gene editing tools to really change that. So lastly, I just want to talk a little bit about the idea of GMO sapiens, or designer babies, and whether this would be something to consider for polycystic kidney disease. And the advantage of doing gene editing at a very early stage in an embryo is that it could be potentially much easier and more effective than doing it in adults. So if you change DNA before the kidneys even form, it's much easier than doing it after the kidney is formed, and you're going to have to change the DNA of every single cell. Now, technologically, this field is moving quickly. Human embryos have recently been gene edited using CRISPR technology in a dish, but those embryos were not implanted, and no designer babies have been born using CRISPR technology, and that was intentional. Scientists are, very cautious not to start tinkering with human DNA until we know that it's safe. Furthermore, I think if you think about a disease like PKD and many other genetic diseases, it's really unclear what advantage CRISPR has over conventional pre-implantation diagnosis technology. So we already have ways of selecting embryos that don't have PKD mutations without even changing their DNA at all. So it would seem to me that that is a safer and more economic approach if we want to have children that do not have PKD mutations. than going in and trying to tinker with their genomes. And this point applies not only to PKD, but for many genetic diseases. When it comes to curing genetic disease, in most cases, PGD is still going to be the approach of choice. And this point has also been made recently in this book that I'm reading called GMO Sapiens, The Life-Changing Science of Designer Babies. Finally, there may be cases where CRISPR or gene editing tools are useful to apply in embryos. There have been cases already of gene therapy or at least cellular therapy in an embryo. But as with anything to do with a child, it is important to consider actions very carefully. And we want to really weigh the ethics of this before going too far with it or trying to, pick children with specific colored eyes and that sort of thing. So that is the end of our crystal ball gazing. And I'll just wrap up with a little conclusions, and then we'll go to the Q&A. So to conclude, I've told you today that PKD is caused by mutations, which are like typos in our DNA and in genes that normally police cyst formation. And we don't yet understand how this all works, how cysts form, why these genes are important, and we still do not have any cure for PKD. Scientists have invented new tools like CRISPR that allow us to edit DNA in cells more efficiently and with fewer errors. These technologies are constantly being improved, and they have not yet been shown to effectively correct disease in human patients. However, gene editing is already employed and is highly useful in the science laboratory. And this is providing an essential testing ground for the future clinical development of this technology in many areas, including PKD. So in summary, I think we're really only at the tip of the iceberg here, both in our understanding of polycystic kidney disease at the molecular and genetic level, and also in our ability to apply gene editing to human beings. But we've had a lot of amazing results just over the last 15 years or so, and this is definitely an area to watch, and I'm hopeful that it will eventually result in major improvements in our ability to treat diseases like PKD. So with that, I'll just say thanks to everyone who's involved here at the UW, also my former colleagues and mentors at Harvard, and the people in my laboratory, and also specifically to all of my funding sources, including the Polycystic Kidney Disease Foundation, which has recently sponsored research modeling human PKD cystogenesis from stem cells, and we're very excited about that. And with that, I will take questions.
Well, thank you very much, Dr. Freedman. We do have several questions. One is interesting about the human genome as the Encyclopedia Britannica, and someone wants to know if the kidney is located on one volume of all those volumes.
Well, it's not quite organized like an encyclopedia. I wish it were. But the answer is that every cell in our body contains the encyclopedia. The encyclopedia is essentially a jumbled mess of genes in no particular order. And these genes are, the cells in our body in different places are reading different entries, different paragraphs in this genome, in this encyclopedia. And how they know which paragraph to read is still quite a mystery. It's the mystery of human development itself. And you can imagine that out of the 20,000 genes that are in this giant encyclopedia, 20,000 entries, each cell is maybe reading about 4,000 of these, but it's a different 4,000 for every different type of cell. And this is not just at the organ level, but every specific type of cell within an organ. So it's a very complex problem, and the kidney, definitely, there are very specific genes that are only expressed in the kidney, but for the most part, the kidney is also using genes that overlap with genes that are expressed in other organs.
We have several questions, Dr. Freedman, that are related to the 384 well plates. Have you begun to use them to actually test potential therapeutics that would affect the growth of cysts on the organoids? And also, are you able to use these 384 well plates to look at mechanisms of action, such as the mTOR pathway and other pathways?
So the 384 well plates with organoids in them are still a work in progress. Certainly something we're interested in pursuing. So the question of whether we're using these yet to look at different drugs, these are costly experiments when you're looking to apply 10,000 different drugs to a set of these plates. So we haven't done those experiments yet. We're starting off with simpler experiments, just seeing if we can make the plates and testing out drugs that we know are maybe involved. And this sort of gets at the second question about the, whether we can look at modifying factors and not just drugs, but pathways. And the answer is yes. I mean, we can certainly use these plates to look at combinations, not just of chemicals, but also what we call silencing RNAs, which are able to turn off specific genes. We can activate specific genes, and we can do this at the scale of thousands of genes, just like we could do it at the scale of thousands of compounds. So I think this is potentially a very powerful technology, and watch this space. Hopefully we'll be able to get some of these experiments going over the next few years.
Yeah, we have a simple question, but I think an important one that relates to how soon CRISPR might be more applicable to human medicine, and that is, what is the estimated accuracy of CRISPR technology? And there was another related question about CRISPR technology. Can this be applied to RNA, as I think is going to be reported soon from UC San Diego?
Sure. So the first question is, what is the efficiency? I said that it's not 100% in the lecture. It's probably, in our experiments, it ranges from about 1% to 10% when you're trying to disrupt a gene. When you're trying to correct a gene, the efficiency can be actually much lower than that. And CRISPR is not the only technology out there. Sometimes other types of gene editing tools have better efficiencies. So a lot depends on what gene you're looking at and in what context. But right now, the efficiencies, I would say, for clinical applications are typically going to be less than 1%, which means that if you tried to do it on 100 cells, maybe only one of them would work. So it's really better suited for a situation where you just need a little tiny bit of something to cure a disease. And I think that there is some evidence that there are diseases out there, I think, like that, where it will be applied clinically first, and that's probably going to happen relatively soon. The second question was, could it be applied to not just DNA, but also to RNA? And the answer there is also yes. I think that the CRISPR, one of the beauties of CRISPR is it's sort of like that search engine, but you can attach to that search function any other type of function that can change DNA or even change RNA. So it's a very powerful tool. People have used it not just to cut DNA and edit DNA, but also just to silence DNA or activate genes. And that's one of the reasons CRISPR is really a special kind of tool. And we're still, as scientists, discovering new ways to use this little search engine for our purposes.
And we have an interesting question related to last month's webinar, and that was about 2 clinical trials that are underway that are repurposing 2 different drugs for type 2 diabetes, metformin and pioglitazone. And the question is whether you can use your mini organoids as a means of testing the mechanisms by which these repurposed drugs might work to decrease the rate of cyst growth.
Absolutely. We can use because you can't really easily test mechanisms in a human patient, where the object is to provide them with the best care possible. But it is, the organoids we can experiment on and play with in a very ethically approved way. So we could certainly apply, and for many drugs, we don't really understand that mechanism of action very well. Even if it has an effect in animal models, we still don't know exactly how it's working at the cellular level. So we're optimistic that we could use the organoids as a system to dissect how drugs are working. And as we get better and better at this, we can envision a situation where we culture organoids from a cohort of patients with a specific disease, and we can test them out, test a drug, for example, out on this different cohort and see the variability within the cohort to better understand what we might get out of a clinical trial. I should also point out that these organoids, we've shown in the Nature Communications paper, as well as in the Nature Biotech paper, that they can actually be used as surrogates for nephrotoxicity. So if we treat a kidney organoid with a nephrotoxic drug, we've seen upregulation of specific biomarkers that indicate that there's a nephrotoxic event. And this may help us predict which drugs are likely to have negative side effects in the clinic.
And so a related question that gets more to basic science, do you think you'll be able to use these organoids to measure electrolyte transport and water transport into the cysts in the PKD phenotype to better understand the molecular mechanisms of the disease?
It's a really great question, and I'm very hopeful that we will. We're certainly investigating this right now, and we've already been able to find that the organoids can transport small molecules from one side of the tubule to the other side of the tubule, and from inside a cyst to outside the cyst. So we're really interested in looking at this, and one of the things we're doing here at the UW to study this better is to develop technologies where we can flow liquid through these organoids and actually watch and see how the tubule or the cyst is processing those fluids. And the idea here is that we may be able to understand better mechanistically why fluid is accumulating in polycystic kidneys.
And another very interesting question, as you mentioned, the encyclopedia is in every cell except perhaps for platelets and red blood cells, etc. PKD, although kidney plays a prominent role, is a systemic disease involving other cell types. So do you envision this therapy as perhaps applying to endothelial dysfunction and other aspects of the disease, such as hepatic cyst formation?
So for the organoids, one of the neat things is that we can create different types of human tissues, not just kidney. We could also potentially look at, we could derive liver cells. In fact, we have done so with stem cells from patients with PKD, and we've seen differences from normal cells without mutations. We could derive tissues like cardiac, which are very difficult to obtain from living patients, and study how PKD mutations are affecting the cardiovascular system and endothelial cells. So absolutely, the other organs affected in polycystic kidney disease is something that our lab is interested in studying, and this is really a black box in terms of human biology. A lot of these haven't been studied. In terms of gene editing, every organ is going to be a little different in terms of how the therapy, the gene therapy, could be administered. So you have to think about delivery to each organ. But we would envision that any gene therapeutic would be administered probably at the systemic level. You know, if you put anything into the body, it's going to move around. And potentially there, too, gene therapy could be targeted to any organ in the body. So I think that there is, potential to really investigate and potentially down the road to treat these other organs and not just the kidney.
And this would be the last question as we're nearing the bewitching hour, and more of an existential question, I guess. How do you view the progress that's been made since the discovery of the genes responsible for PKD, and how do you view CRISPR technology and similar technologies in getting closer to an effective treatment, if not a cure, for PKD? Our patients are obviously very anxious to hear what kind of timeline you envision.
Well, I'll put it to you this way. We are, in my view, we've made more progress in the last 30 years than on PKD than we had in, or at least in the last 100 years on PKD, than we had in the previous, 100,000 years of human history. So I think that we are just at the beginning of the log phase of exponential improvements. And the speed, we are going to eventually reach a point where technology can cure these diseases. That much I am convinced. The speed at which we advance up that log scale will depend on the investment our society makes into curing these types of diseases. So I think that it is a matter of time. The timeline can be certainly accelerated and I think it's great that so many people have tuned in, and what we need is for this type of discussion to really go mainstream and for the interest and resources of our society to be dedicated to curing these diseases in the most rapid time frame possible. If we put a Manhattan Project together to cure diseases, it will happen more rapidly. With the pace of our current technology, I think we're probably, looking at still slower but nevertheless steady progress towards newer and better cures. So I think there's a lot of reasons to be hopeful, and I think everyone's involvement is only going to hasten the pace of this.
Well, Dr. Freedman, thank you for an exciting and illuminating webinar. Alexis will follow with a few housekeeping details and what we can look forward to in future webinars.
And I just wanted to make sure everyone noted that I got Benjamin's name correct again after missing it the first time. Just to draw your attention to our next month's webinar Wednesday, April 20th, we will be talking about total kidney volume and PKD. So tune in while David talks about what total kidney volume is, why it's important, how it's measured, and kind of just answers all your questions concerning that. And then also don't forget about the PKD National Convention 2016. You'll be able to meet Dr. Freedman at the convention. He'll be joining us to talk about the work that he's doing, and so don't miss your opportunity to meet him in person. And that is June 24th through 26th in Orlando. And you can find out more at pkdcure.org/convention. As always, thank you so much for your time, and we hope you have a wonderful day, and look for the email to come in the next few days with the survey to let us know how we did. Thanks a lot, everyone. Thank you, Dr. Freedman, and everyone have a great day. Bye-bye.